Hello everyone, my name is Jiaming Wang. In this talk, I will tell you about our work towards practical and scalable molecular network. Nowadays, researchers are able to build micro and nano implants that can sit in the brain, travel through the bloodstreams to perform target drug delivery, all the way to contraception implants and lab-on-a-chips that can conduct in-body diagnosis. However, the question remains, how can we communicate and network such micro and nano implants? Traditional implants like cardiac defibrillators and pill caps use wires, which may not be suitable. These wireless implants have a relatively large form factor. In some cases, they require an external device to power up. Besides, wireless signals does not propagate well inside the body. Instead, researchers have been turning into a new paradigm, molecular communication, which is inspired by the chemical signaling between cells inside the body, like the neurons release neurotransmitters or the glands releasing hormones. In its simplest form, an implant can transmit best by releasing particles in the bloodstream, so releasing molecules for bit 1 and nothing for bit 0, which is similar to on-off keying. The molecules then travel along the blood flow and arrive at the receiver. Unfortunately, a lot of the work on molecular networking is still theoretical. There's very limited experimental work there. Researchers build testbeds to emulate the molecular channel, such as using niacin base, salt molecules, or magnetic particles. At the same time, there's also research in synthetic biology on building transmitters and receivers, but they do not focus on the communication channel. Considering this, we need more experimental research to understand how to build practical molecular networks. In our prior work, we focused on understanding the molecular channel. In particular, the channel is based on diffusion. If you focus on the released particles here in red, you can see that as they propagate, the color diffuses, the channel stretches, creating long tails and high intersymbol interference. We show that by accounting for the complexities in the channel, we can significantly improve the decoding for a single transmitter. But in today's paper, we ask, how do we scale the molecular network from a single transmitter to multiple transmitters? Past theoretical work proposes three types of media access protocols. The first is TDMA, which is great in theory because it avoids interference, but in practice, it requires synchronization. Let me tell you why synchronization is hard in molecular channel. As comparison, in wireless channel, the signal propagates at the speed of light, so it takes only 100 nanoseconds to travel 30 meters, making it easy to synchronize. However, for molecular channel, blood flow speed is only about 1 meter per second. Thus, it will take the signal 1 second to travel only 1 meter, where synchronization is much harder. But more importantly, is that unlike wireless, the molecular channel is asymmetric. Molecules cannot propagate back against the blood flow, so you do not get feedback from the receiver or a trigger to tell the transmitters to transmit. The second approach is molecule division multiple access, where each transmitter uses a different type of molecule. This would remind you of FDMA, but instead of different frequencies, we use different molecules. However, with this approach, you cannot scale because the receiver grows way more complicated to measure different types of molecules. And finally, there is CDMA, where we can use a code to avoid interference without needing synchronization. However, we cannot apply CDMA as is, since molecular signals are non-negative and have very high ISI. So, how do we design an efficient medium access protocol for molecular networks? In this work, we propose MoMA, Molecular Multiple Access, a practical and scalable protocol to enable molecular networking with multiple transmitters. Here are the key design principles of MoMA. First, we account for the unique channel properties including high ISI, lack of feedback and synchronization, non-negative signals, etc. We customize CDMA codes for the molecular channel to allow us to separate different transmitters. In the meanwhile, we keep the transmitters as simple as possible. They can transmit anytime they have data. We just let them collide and push the complexity to the receiver. Thus, at the receiver end, we can now nicely separate different functionalities like packet detection, channel estimation, and decoding. we'll have to do them jointly and iteratively. Later, I'll tell you about the importance of packet detection, which were overlooked by previous work. And finally, we show that you can improve the performance by using 2 or even more types of molecules. I won't have time to go over everything, so I'll focus only on 2 ideas. First, let me tell you why packet detection is so important. Without the synchronization among transmitters, the packets can arrive at any time. So while decoding, we'll have to keep checking if new packets arrive, which is very difficult when we're already dealing with other packets. If we miss a single packet, the bit error rate explodes for every packet on hand. Here's the result, where we intentionally make 2, 3, or 4 transmitters collide. You can see that if we ever miss detecting a single packet, the bit error goes from almost 0 to more than 30%. So why is it not easy to detect new packets? Prior theoretical work in molecular CDMA set the coded molecules for bit 1 and nothing for bit 0, resulting in the unstable concentration of molecules in data symbols. This is extremely problematic when another packet arrives and exactly fills the concentration gaps. Adding the higher intersymbol interference, it becomes very hard for the receiver to distinguish 2 transmitters from just 1 transmitter sending bit 1s. So what does MOMA do? We make sure that the pattern in preamble is very different from data symbols. First, instead of sending nothing for bit 0, MOMA sends the complementary code, so the molecule concentration in data symbols becomes balanced across time. For the preamble, however, we do the opposite. We repeat the chip once back to back to repeat large fluctuations in concentration, which allows us to more easily detect new packets. Let me show you how the signal looks like in an experiment when you do this. You can see that although the 2 signals share equal average concentration, the blue preamble changes way more drastically than the yellow data. And as a result, the new packets can be easily told from the increased signal power as well as its fluctuations. Another highlight is how MOMA makes better use of multiple molecules. Suppose we have 4 transmitters and 2 molecules in the system. What you could do is to divide the 4 transmitters into 2 groups, and the first group uses molecule A, while the other uses molecule B. Then you can use CDMA within each group to separate the transmitters. Using this scheme, naming molecule division CDMA, you would reduce interference by half. But what MOMA does is to have all transmitters use both molecules. Feel confused? Although this idea might be counterintuitive at first glance, because collisions are still there. But there are more advantages. First, MOMA can send independent streams of data on different molecules. Thus, you can use longer code while keeping the same data rate. But remember, longer code means much lower bit error rates. More importantly, you can achieve much better channel estimation and packet detection. Remember, we're saying how important packet detection is. The longer code plus individual preambles on each molecule means much more samples for these 2 procedures. This turns out to be way more important than just reducing collisions. Let me show you some results. MOMA is evaluated on an emulated testbed built with pumps and tubes. The evaluation includes 2 different topologies, the long one and the fork one. The dataset also includes 2 different molecules, which are the table salt and baking soda. We compare MOMA with the baseline molecular division CDMA, which was explained a few slides ago. The network has 4 transmitters and 2 molecules, while the data rate per transmitter are the same for both protocols. We can see that when there are only 1 or 2 colliding packets, it does not really matter. But when it comes to 3 or 4 colliding packets, MOMA achieves 70% higher throughput than the baseline, and it is only 10% loss compared to the data rate. There are much more results in the paper, but in the interest of time, let me conclude here. First, molecular networks are essential for communicating with micro and nano implants. Second, we propose MOMA, which is a MAC protocol that can scale to multiple molecules while keeping the transmitters as simple as possible. But in the meanwhile, it also has several limitations, like it is only uplink and assumes a complex receiver. But for future work, we do need a lot more networking research in this field. We need to rethink and redesign our networking protocols while working closely with biologists and bioroboticists. For more details, please refer to our paper and artifacts. Thank you.